very unusual circumstances. We are recording this, so people who are not on right now on live can watch this online. In a couple of weeks, we're going to post it. My introduction to Joel is going to be very brief because I hate to take even a minute of his time for his presentation. So we welcome especially our students. We hope that the students, instead of going to Gentle Ben's, are online and are listening, because this is going to be a very interesting presentation, as you can tell from the title. Also want to introduce and welcome our SEAC volunteers. I've seen a couple of emails running around from them, and I hope that some of them are listening in and viewing this presentation. Finally, welcome to the faculty and staff of SEAC. I hope you're all well. I hope you're all staying healthy and not drinking any bleach. So Professor Cuello is a professor in biosystems engineering. He has a wide variety of interests, going all the way from the technology of greenhouse and especially vertical farming in greenhouses, to now what looks to me to be a kind of adventure for him that is merging the quest for medicinals in medicinal plants, specifically in hemp at this point, and combining that with cell culture technology, which is designed to provide clones of plants that have specific characteristics. I think that Professor Cuello's outlook on life is all summed up in his website logo, which is called Bioimagineering. And that's what he does for a living. He's combining biology, plant biology, other kinds of biology, with imagination and engineering. So here he is, Professor Joel Cuello. Welcome to SEAC seminars, and it's up to you now.
Thank you so very much, Rafi, and thank you, Murat, as well, for having me today. And I'd like to say good afternoon or good evening, it's still early, I suppose, to everyone, and thank you for joining us today. So I'll be talking about, as the title says here, optimizing hemp cultivation, cell culture versus open field cultivation. I'll just go through the presentation, and I look forward, of course, to have an interaction with every one of you in terms of questions and answers after this presentation. So we're talking here about industrial hemp, and first let's clarify what is the difference between marijuana and industrial hemp. As you know, they are of the same species, which is Cannabis sativa. Well, the distinction is that I'm going to use today is a legal distinction as defined by the U.S. federal legal definition. So if the hemp plant has a concentration of THC, which is tetrahydroxycannabinol, that's the psychoactive compound found in marijuana. If the THC level is less than 0.3%, then that plant is classified as industrial hemp. If the THC concentration is higher than 0.3%, then the legal definition of that plant is that it is a marijuana plant. And today, most cannabis or marijuana plants or strains or cultivars, they have an average concentration of about 12% THC. But we're talking here about industrial hemp, whose THC level is less than 0.3%. But it is of interest to a lot of producers, a lot of farmers, because of the multifarious uses of hemp. including the different uses for its vegetative parts of the plants, but also because of the rich pharmacopoeia or the cannabinoid compounds that are found in the industrial hemp. So you can think of industrial hemp as a chemical or a biological factory. And of course it produces about, now it's been known to produce about 113 different phytocannabinoids, or those natural compounds, one of which is CBD or cannabidiol. And there's been a number of clinical research studies that have been conducted, and the initial results of those research studies is that they have a lot of benefits, which I will show in the next slide or two. But this is for green wellness, and the CBD market, because of this known beneficial effects of CBD, has really generated a lot of interest in the market. So by 2021, the CBD market is projected to be worth $200 billion. And in 2024, it is projected to be worth about 24 billion US dollars. So it's not surprising that there's a lot of producers as well as manufacturers that are interested in industrial hemp production. So again, as to why this is generating a lot of medical interest is that, again, preliminary results of some clinical research has shown that CBD shows potential for the treatment of a number of conditions. And I've listed here a number of those: addiction, aging, anxiety, obesity, depression, Parkinson's, and post-traumatic stress disorder. But what I would like to emphasize is that there's still a need for high quality evidence to show the effectiveness of CBD as treatment for any one of these conditions here. And hopefully that is going on currently. So as you're all aware, industrial hemp is made legal in a number of states here in the US, including, of course, here in Arizona, and that happened last year. And of course, the US Department of Agriculture has a set of requirements that are meant to be complied to or satisfied by the different states, including Arizona. And I listed here just 4. This is not a complete list, but of course, there needs to be a tracking of where the hemp plants are being grown, provisions for testing the THC levels, because again, the federal government considers that to be quite crucial in terms of defining what is a hemp plant and what is a marijuana plant. Disposal of non-compliant or hot plants. These are plants with THC levels of greater than 0.3%. And provisions spelling out what compliance looks like, the penalties and sanctions for negligent violations, and standardized inspection procedures for all farms growing hemp. So I know that the local USDA here in Arizona has had a lot of workshops for growers and producers, informing them of what these compliance regulations are. So pretty much all the producers are up to speed. So in this talk here, I'd like to make a contrast between open field cultivation of hemp plants versus the alternative production systems for hemp plants, particularly cell cultures. So the thing about open field cultivation of hemp plants is, as you know, the plants are subject to variable conditions of the environment. So that would be temperature, relative humidity, amount of sunlight, and so on and so forth. So that's why I highlighted there the climate and the weather. But it's also dependent on the variety of the particular industrial hemp plant, the sex, the age, development stage, cultivation methods, time of harvest, storage conditions. So all of these factors are very critical in terms of determining the growth rate of the plant and the productivity of the plant, as well as how much cannabinoids the plants produce, and even what set or types of cannabinoids the plant will produce. So that would influence directly the CBD productivity of the plant, and also the THC level. The first batch of industrial hemp production here in Arizona took place, I guess, late last year and over to the beginning of this year. And unfortunately, a lot of those crops ended up having THC levels of greater than 0.3%. And so by law, they had to destroy their crops. So that's really unfortunate for the farmers. And that is one of the hazards of growing industrial hemp plants in open farms, because you won't know, you can't guarantee that at the end of the production cycle, your THC level is going to be below 0.3%. Particularly that we are in a semi-arid environment where we have some extreme weather conditions in terms of temperature, in terms of light level. The impact of these extreme conditions is that they tend to overproduce secondary metabolites, and that includes THC, because THC is a secondary metabolite. So just a note on that, you might broadly classify plant chemicals into two groups. One is primary metabolites, the other one is secondary metabolites. Primary metabolites are carbohydrates, lipids, proteins, nucleic acids. So these are chemicals with known direct roles in growth and metabolism of the plants, photosynthesis, respiration. But secondary metabolites, this is a whole range of chemical compounds found in plants that do not have any known direct roles in growth and development. So they do not have any direct roles in terms of respiration and photosynthesis. However, the plants produce them, and typically they are produced by the plants as part of their defense mechanism. So for instance, when a leaf of a plant is encroached upon by certain microorganisms, the cells to which the microorganisms get in contact with, they will be coaxed to produce these secondary metabolites. And the purpose of producing those metabolites is really to kill the microorganisms. So that's an example of the defense mechanism that plants have. And of course, different species of plants, in fact, different varieties of plants will produce different sets of these secondary metabolites. And that is why scientists have been mining the secondary metabolites for a long time now from plants, because they have a lot of excellent and profitable commercial uses, such as pesticides, flavors, colorants, even pharmaceuticals, of course. And so CBD and THC and other cannabinoids, they are secondary metabolites. So they're very sensitive to these physiologically stressful factors, including changes in environmental conditions. So I was not surprised at all that in the initial trials of open farm cultivation of industrial hemp, that there's a number of cases where the plants ended up having THC levels of greater than 0.3% because of the stressful conditions in a semi-arid land like Arizona. So this is a picture that I took earlier this year at Yuma Ag Center. So this is a University of Arizona facility where they are growing industrial hemp for research purposes. And of course, they're using drip irrigation here. So that's a closer view of the industrial hemp plant. So of course, it looks like marijuana because it's the same species. I'd like to introduce Yasser Medipour. He is a research scientist in this funded project that we have in terms of developing cell cultures for production of CBD in industrial hemp. So this was the workshop in Yuma that I participated in, where I gave an invited talk last, I believe it was February. Okay, so let's talk now about alternative methods of cultivating hemp. If it's not going to be an open field cultivation, what are the alternatives that are available to us? So the quickest alternative, of course, most convenient would be either a greenhouse or a warehouse. So this is Wheatfield Gardens in Niagara County in New York State. And this is a greenhouse facility where the company is growing, of all things, lettuce plus industrial hemp. So this is a nighttime view of their facility, and obviously they're using red LEDs. That's why you have that red glow emanating from the inside of the greenhouse. So this is an interior view of their facility where they're growing vegetables, and at the same time they're growing industrial hemp. And they're growing it hydroponically. Okay, so if it's not a greenhouse, then of course you can use a warehouse type of closed environment where you can be more precise in terms of your regulation and control of the light level, as well as the other environmental factors, including relative humidity, temperature, the air velocity, and so on and so forth. Okay, then in support of either an open field cultivation or a warehouse type of cultivation or a greenhouse type of cultivation, we have this biotechnology technique that's been available for some time now, for decades. Plant tissue culture, which is also known as micropropagation, to be able to support that type of production. So what is plant cell culture or plant tissue culture or micropropagation? It's a technique for the rapid micropropagation or rapid production of shoots or plants. And these plants are true to type, meaning to say that they are clones. They are the same genetically, and because they're true clones, then they have the same morphological characteristics as well. So micropropagation is a rapid method of multiplying these clones in vitro, that is in glass or in a controlled environment vessel. There's something amazing about plants, and that is in terms of their malleability and plasticity. And you can manipulate plants easily just by regulating the relative concentrations of these two major plant hormones, cytokinin and auxin, in their growing medium, in a cell culture or tissue culture setting. So in general, if the ratio of cytokinin to auxin is greater than 1, you're only going to be producing shoots, meaning to say the upper parts of the plants. You'll be producing stems and the leaves, but you're not going to produce roots. If we go down here, if the cytokinin to auxin ratio is less than 1, what you're going to have are only roots. You're not going to have the shoots. You're not going to have the stems. You're not going to have the leaves, but you're just going to be growing roots. And if the cytokinin to auxin is about 1, what you'll be producing are undifferentiated cells. This is equivalent to stem cells in mammalian cells. It's very easy to produce or generate stem cells or undifferentiated cells for plants, and then convert those undifferentiated cells into shoots and roots and even flowers or fruit. With plants, that's very easy to do. And it's just requiring some manipulation of the hormones. But of course, with mammalian cells, including human cells, that's a tough thing to do. If you're going to coax the production or the generation of specific organs or tissues out of undifferentiated cells. But scientists, of course, are still looking into that. So let's consider this case where the cytokinin to auxin ratio is greater than 1. And then we're going to be producing shoots, and this is the principle used in micropropagation. So the rapid multiplication of the shoots takes place in vitro inside these aseptic containers, and they are in this growing medium, which is a solid medium because they added agar, where the plant hormones are present, together with all the other essential nutrients. So this is what I actually did for my master's in agricultural engineering back at Penn State, in the early 1990s. So I did micropropagation, and this is for rapid multiplication of an ornamental plant. But we went further in terms of regulating the various environmental factors that regulate the process. So we controlled the temperature, we controlled the light level, and we also regulated the CO2 concentration in the air that is being channeled into these closed vessels. We got some good results as well. So thanks to Rafi, who forwarded to me this article a few days back. about a company called Segra, which is based in British Columbia in Canada. It's a micropropagation company for cannabis. I believe this is for marijuana and not for industrial hemp, but it's a similar process. So what they're doing here is that they're multiplying the shoots that are genetically identical, true to type, which are meant to be really high yielding types and high quality types of marijuana cultivars. And so they just had a press release saying that they struck an agreement with a licensed producer in Canada where they are producing about or supplying about 40,000 plantlets. And to their knowledge, this is the first ever large-scale deployment of cannabis plants that are directly derived from tissue culture or micropropagation. So that's great for them. That's wonderful for them. And of course, that's the technology or innovation is directly transferable to industrial hemp as well. So regardless of whether the production system is open field or in a greenhouse or in a vertical farm setting or warehouse setting, of course, it matters that you have high quality X plants to begin with or to start with. And so the service of this company certainly is valuable in that regard. So the alternative cultivation method that I really would like to focus on today is not open field, not greenhouse, not warehouse or vertical farm, but organ cultures and cell cultures. So let's start with organ cultures. Again, if you manipulate the relative concentration of these two plant hormones, cytokinins and auxins, one possibility is the cytokinin to auxin ratio is less than 1, and you will end up with just roots. And this is what I did for my PhD as well as my second master's at Penn State, where we worked only with roots because the chemicals that we're interested in are biosynthesized in the roots. So it didn't really make sense to grow the entire plant because that would be very energy inefficient and wasteful because you don't need the leaves, you don't need the stems. You're only interested in the roots because the chemicals are biosynthesized only in the roots. And so we developed this production system here where we were able to generate just roots. These are different species, obviously. And of course, my interest in my PhD was to develop the scale-up production system for these roots, and that is in terms of bioreactors. So this is an ebb and flow bioreactor that I developed for my PhD dissertation, and I was able to demonstrate successfully that yes, these roots can be grown successfully and competitively in a scalable system such as the ebb and flow bioreactor. All right, but this is applicable if your chemical of interest is produced in the roots. Now, in the case of industrial hemp, CBD, even THC, are not produced in the roots of the industrial hemp plant, but in the trichomes, and that is part of the fluorescence organ of the plant. So producing roots probably is not going to work. However, in an industrial hemp, there are, as I mentioned, about 113 different cannabinoids. And scientists are very interested in terms of the medical application or effects of these cannabinoids. So it might be, we don't know yet, it might be that some of these cannabinoids are actually present in abundant concentrations in the roots. And in that case, this would be a production technique or strategy that would work very well for those particular types of cannabinoids that might be produced in the roots. But today we're focused on CBD, which is, again, produced not in the roots. And so I'm presenting here another approach, which is cell cultures, to be able to produce the CBD, in a bioreactor as well. So as I mentioned, if the cytokinin to auxin is generally equal to 1.0 in terms of the ratio, that's when you produce the undifferentiated cells. So this is a picture of undifferentiated cells. And we actually took this in my lab years ago when I had a PhD student working with cell cultures for Agave Pacifica. So these are actually cell cultures of Agave Pacifica. So these are undifferentiated cells. They're not leaves, they're not roots, they're not shoots, not any type of organ, but just cells. This is a mass of cells that are growing like this. But the idea here for the industrial hemp is to be able to isolate the right tissue, the right cells that actually produce the THC or the CBD in this case, and then be able to proliferate those cells in vitro. Because then if you're able to mass produce those cells, you do not have to grow the entire plant anymore. You don't have to expand the energy that's needed to grow and to produce the roots or the leaves or the stems. You only have the cells that you need that will actually manufacture the CBD. And in reality, there's a number of groups now that have successfully generated cell cultures for industrial hemp and for the cannabis plant. So this is not completely new. You can generate cell cultures from industrial hemp. The real challenge is having a cell culture strain that will optimally produce CBD. That is the real challenge now. Because some of these groups, yes, they were able to successfully produce the cell cultures, but they have virtually no production of CBD. And of course, that's a problem if what your interest is producing the CBD. So that is essentially the goal of our research that we will be doing as soon as we're able to use our lab for this purpose, is to be able to generate the cells in an appropriate manner and using the right cultivars and using the right environmental conditions, so that we're not only able to successfully generate the cells and grow the cells, but that these cells actually produce CBD. But we're very optimistic in terms of the particular strategy that we're aiming to implement. So this is just a picture of the fluorescence of the cannabis plant. And if you see these white things here, those are the trichomes. And those are the tissues that actually produce the CBD from the plant. That's another micrograph highlighting the structures of the fluorescence of the cannabis plant. So in terms of cell cultures, you grow them just like any other cell cultures. You need to provide light. It doesn't have to be sunlight. It can be, it's better actually if you can provide it using artificial lighting, such as light emitting diodes, because then you can really regulate not only the concentration and the photo period, but you can regulate as well the light quality, you know, how much red, how much blue, maybe a little bit of UV, and definitely you can design the light quality combinations so that you can elicit more of the CBD from the cells. The same thing with the nutrients, nitrogen, phosphorus, potassium, and others, of course, water, air, and of course, CO2. This is a photosynthesizing set of cells, and they need CO2 as an input for photosynthesis. So certainly CO2 will have an impact in terms of their growth rate and productivity. So the great thing about cell cultures is in terms of the industrial scale up of this production system. So in the lab, we grow the cells in flasks or test tubes or bottles. That's fine. But if this is going to be a commercial operation, you cannot produce them in millions or hundreds or multi-million containers, because that would be extremely inefficient, labor-wise, as well as in terms of just the production of the plants. So the approach, of course, the technique in industry is you grow them in scalable bioreactors. And this is our Accordion Bioreactor that we ourselves developed originally in the lab, and that the U of A has the patent for. And of course, these types of bioreactors can be setup in a greenhouse type of structure or even a warehouse type of structure. So these are the major advantages of producing CBD from hemp cell cultures. There's no need for arable land because you're not going to be growing plants in soil, so there's no need for arable lands. Water savings, certainly. With open field cultivation and even with greenhouse, there's significant transpiration taking place. So there's a lot of usage of water by the plants, but with cells, that is dramatically reduced. Nutrient savings, you're not fertilizing in an open field, so there's no leaching of nutrients into the groundwater. The nutrients stay in the liquid nutrient solution inside your bioreactor, and you can just circulate it until the nutrients are consumed by the cells. It's weather independent, so it doesn't matter if you're here in the Southwest or you're in Norway or the Middle East. It really doesn't matter because you're able to control your production environment. No pesticides, no herbicides, no assimilation of toxic substances from the soil, clean and safe production, consistent and reliable production, and it is scalable. And of course, the great thing about this is once you have tailored your set of environmental factors and nutrient factors such that you have defined the optimal set of conditions, not only for the growth of the cells, but for the production of the desired compound by the cells, in this case, CBD, then you establish it. All you need to do is implement that set of conditions every time you grow your cells in the bioreactors, and you will have exactly the same results every time. So it makes your production really reliable, predictable, dependable, which is great. That's all you want really when you have a large-scale production system. Is you want to be predictable, you want to be dependable and reliable in terms of what you will have. And in this case as well, you eliminate that concern, that problem that let's say you grow your hemp in the open field and you wait for weeks, and in the end, you will have to test for the THC level of your crops, and you're sweating and nervous, hoping and praying that the THC level is going to be below 0.3%. If it's below 0.3%, that's fine. But if it happens to be above 0.3%, you're going to have to destroy your crops. And that is just, that's frustrating and beyond frustrating, of course, for the growers and the producers, because that is basically a commercial loss for them. All right, so in regard to bioreactors, so this is the greenhouse facility at the U of A on top of the 6th Street garage, where we have, our lab has a facility there, and we have this setup of the Accordion Bioreactors, which we've been using for growing algae cells, but it's exactly the same thing. These are cells. So imagine this to be the hemp cells, and they will look exactly the same because they will be photosynthesizing as well. So you can grow them efficiently and productively in these bioreactors. This is just a time lapse that's showing you from day 0 where we inoculated the bioreactors with a certain amount of cells, and that's why it's light colored. And as you progress over time, you can see that the intensity of the greenness of the culture is increasing, and that's because the cells are growing in the bioreactors. And that's just the growth curve. And we've developed strategies so that you can maintain this peak production of the cells, so that you're not going to be wasting time, but you will always be operating at the maximum growth rate of the cells. We have different types of bioreactors that we develop in the lab. This one is the regular Accordion Bioreactor, where the liquid nutrient solution in the cells is recirculating. So it's going into the bioreactor vessel, it will go out, and then it will go back in again. So it's recirculating that way. It's advantageous in terms of the mixing of the cells. However, this might be a concern because this is a higher stress type of environment or higher shear stress environment of a bioreactor. And so we developed the Accordion Bioreactor where the liquid and the cells stay inside the vessel. It doesn't recirculate. It doesn't go outside and then come back in. It just stays inside the vessel, and you have an aeration here that produces the mixing, the hydrodynamic mixing inside the bioreactor. And of course, the air could be mixed with elevated levels of CO2, which would favor greater growth rate for the crops because of improved photosynthesis. These are some of the advantages of these bioreactors. And it's possible, of course, to grow the crops photoautotropically, meaning to say that its growth is exclusively dependent on the photosynthesis, or it could be heterotropic. If you don't want to grow them photoautotropically or by photosynthesis, you can grow them completely in the dark, and the sugar then is supplied exogenously. So you add sugar or some form of carbon into the medium, so this becomes a type of fermentation process. The advantage of fermentation is that the cells grow much, much faster as when they're being grown autotropically. On the other hand, you really have to be operating this under aseptic condition, because when you have sugar in the liquid medium, bacterial spores in the air would be attracted to that, and so the bacteria will be growing together with your cells. And typically the bacteria win and they just overtake the growth of the cells. Mixotropic is combination of autotropic and heterotropic production. So these bioreactors that we developed accommodate any of these strategies for cell production. This is the third type of bioreactor that we developed. It's the Accordion air loop bioreactor. We have a fourth one, the axial dispersion bioreactor, and we have a number of other bioreactors that we have developed or in the process of developing. So again, here's a picture, a close-up of the fluorescence of the cannabis plant. And again, I just would like to reiterate the advantages of being able to develop cell cultures of hemp that actually produce the desired compound, because then the industrial or the scaled-up production of that system is far, far more convenient and controllable and predictable and dependable compared with open field cultivation. So I'd like to acknowledge our sponsor, that's Taurus Ag LLC, which is based in Phoenix. And I'd like to acknowledge Yasser, who is the research scientist working on this project. So that's my picture at the Yuma Ag Center farm back in January. And with that, I'd like to end the presentation and we can go into the Q&A. Thank you so very much.
All right, Joel, thank you. Thank you for your nice presentation. We have some questions for you from our audience, and before I get to the questions, I would like to also thank everyone joining us. This is Murat Kusira. Let's get started. Joel, I think the first question is, can micropropagation be used for other plants besides hemp?
Yes, absolutely. And historically, micropropagation really was developed for other plants. And it's only recently that the technique is being considered for application for hemp or for cannabis. So for instance, micropropagation has been used for ornamental plants, for fruit plants, for cereal like rice and wheat and barley, basically all types of plants now. Orchids, for instance. So yeah, it's been applied in many, many types of plants.
All right, great, yes. Another question, what other secondary metabolites have commercial potential in hemp? I believe you mentioned several metabolites, but the question is, what are some of those secondary metabolites having commercial potential in hemp?
Right. So that's a great question. I'm sure that the hemp plant has different types of metabolites, but it's been focused on the cannabinoids. And there's about 113 of these cannabinoid compounds that have already been identified. And the hypothesis is that similar to THC and CBD, the other 111 cannabinoids would have a lot of medicinal effects on humans, similar to what CBD and THC is doing. But there's research going on right now, but there's not a lot of actual results that have been put out yet. But I'm sure that we are expecting that we'll be seeing a lot of those in a short period of time, given the high level of interest in those cannabinoids.
Okay, great, Joel. Yes, the questions started coming. We have more questions in relation to the first, the previous question. So we have the secondary metabolites. We are targeting them to produce more, maybe, or control them actually. So you mentioned some techniques or systems, hydroponic systems actually used to grow hemp. Are there procedures to control the percentage of secondary metabolites? Assuming that we understand, we have sufficient understanding of this plant, and then we can control or manipulate the plant actually, or its environment, to control the secondary metabolite concentration.
Yes, yeah, absolutely. So with again, THC, CBD, as well as the other cannabinoids, these are secondary metabolites, and in general, plants mass produce the secondary metabolites when they are physiologically stressed. So if the goal is to maximize their concentration in the cells or the plants, then it would be good to develop a growth regime where they are subjected to specific stressful physiological conditions. And that could be in the form of intense light level, or maybe a bit of an elevated temperature, or nitrogen deprivation in the nutrient solution. These are typical techniques that are being used in terms of eliciting secondary metabolites from plants. And they could be applied definitely on cannabis plants to be able to coax the overproduction of these secondary metabolites.
All right, Joel. What are the optimal conditions or set points for micropropagation systems and settings in terms of CO2, light intensity and quality, and humidity?
Right. So that would depend on the plant species that you're growing. So for my master's, I worked with ornamental plants. One is, it's called Buddleia alternifolia, which is Egyptian henbane. That's not very familiar. But the second one was chrysanthemums, so that's more familiar to the US audience. I can't remember exactly the levels of light that I used, but we were able to define, we published this, so we were able to define the range of temperature, the range of light level, and the concentrations of CO2 that resulted in number one is the growth rate of the chrysanthemum plantlets, as well as the number of shoots that were produced, because that's critical in micropropagation. You want to multiply the shoots so that you end up with more plants or plantlets. So those are the two basically dependent variable that we focus on.
Okay.
And by manipulating the environment, we were able to maximize or optimize those dependent.
Okay, Joel, thank you. Okay, so here's another question. Is it possible to make undifferentiated hemp cells being differentiated to trichomes for trichome culture to produce cannabinoids?
So that's part of our objective, actually.
All right, okay.
So in all honesty, the answer is, I don't know. No one knows at this point, but that's what we are aiming to do.
All right, okay, Joel. How is the harvesting process in cell culture methods inside the photobioreactors?
All right, that's a great question. So, well, that's actually a question, a big question in the algae industry. So one way is using centrifugation, but centrifugation is a process that requires a lot of energy. It's power intensive. So if you're just going to rely on it, it's probably not going to be very efficient, commercially speaking. So what we do is the way that our bioreactor is set up is that we have this opening at the bottom. And so we can stop the production once it's reached its desired biomass level. And what happens to the algae cells is that they settle out near the bottom. And then that's when we open the bottom of the reactor, and so they drain out. So what we have there is a far more concentrated algae solution or mixture, if you will. And then we then use the centrifugation to further purify that and concentrate the cells. In that regard, the use of centrifuge is not that power intensive anymore because the input into the centrifuge is a highly concentrated solution of algae.
Okay. All right, great, Joel. I think somewhat related to what you described is this question, and this is I can tell this is an engineering and systems related question. Can you elaborate on the volume of water used in bioreactors and the amount of energy used to dewater the final mixture?
Right. I don't know the answer to that yet because the focus that we had, at least with the algae culture, is really in the production as opposed to the downstream processing, including harvesting. So we didn't really measure how much energy we were using in terms of harvesting the cells.
Okay. Let's see. I'm looking at the Q&A window here. Can you get rid of pathogens in plants with tissue culture process?
Oh, okay, great. Yeah, I forgot to mention that. So one big application really, or the interest in micropropagation or plant tissue culture is to be able to produce cultivars that are virus-free or disease-free. So that is one principal application of tissue culture or micropropagation.
Okay, I see. Yes, great, great. Okay. Next question: how do the companies that have successfully created cell cultures with the phototoxicity of cannabinoids to plant cells?
Okay, so the ones that have succeeded in producing cell cultures of hemp or marijuana or industrial hemp, these are research groups. These are not companies.
Okay.
So yeah, they're doing this in the lab, or they did their studies in the lab. So these are not commercial companies.
Okay. All right, Joel, thank you. I'm looking at other questions. So, all right, as far as the Q&A window, I think these are all the questions that we have for you, Joel.
Right.
I would like to wait maybe another minute if
Sure.
we have other questions from our audience here.
And Rafi might have a question.
Yeah, Rafi. Rafi always has good questions. Rafi.
Not this time. I think this was really, at least for me, a very interesting presentation because it's open to so many possibilities. And Joel correctly acknowledged that this is at the very beginning, which will make especially medicinal cannabis very precise and very clean, so that medical research on the cannabinoids can take place, in a way that is scientifically meritorious.
Right, right. I'm glad that you mentioned that. So that really is the promise of cell culture production of CBD and other cannabinoids, is that it becomes a high precision method of production. It really is high precision.
Sure, sure. Joel, we have another question for you. What about the phytotoxicity of cannabinoids to plant or algae cells in tissue culture, in tissue cultures?
Okay. Okay. So I use the algae cells growing in bioreactors just to illustrate that we can do the same thing for hemp cells. So I was not making a suggestion that we would be growing them concurrently in the same vessels.
Sure.
So in terms of the phytotoxicity of hemp cells, so they produce some of these cannabinoids that would be toxic to certain microorganisms, but they're not toxic to the hemp cells themselves. Okay. At least, you know, when they're able to regulate the production, and they do, the cells regulate how much of these cannabinoids they produce. So I'm not concerned at all in terms of growing the cells, the hemp cells, in cell cultures, in bioreactors, and them producing toxic levels of metabolites that would be detrimental or that would be lethal to the hemp cells.
Sure.
I'm confident that's not going to happen.
Yeah, yeah. Okay. Joel, you mentioned about stressing the culture and you know, to generate more secondary metabolites, right, and their environmental conditions you mentioned. Among those, which one do you see the kind of most critical or one that would enhance the secondary metabolites?
All right. So definitely light. And when I say light, of course, it could be the intensity and or the light quality, or the wavelength of the light, red, blue, UV. Those have been proven to work in a lot of plant cells in terms of coaxing the cells to produce metabolites. The other one is playing with the nutrients. So let's say nitrogen deprivation. So you just reduce the amount of nitrogen in the liquid medium. And typically that happens anyway, because as the cells grow, they consume the nitrogen that's available in the liquid medium. So as they grow, they will reach a certain stage where there is a limiting amount or concentration of nitrogen in the liquid medium. So perhaps at a certain point, you stop replenishing the nutrients in the liquid medium and allow the nutrients to become limiting. And that in itself will also put the cells in a type of physiological stress where they would be elicited to overproduce certain metabolites or cannabinoids in the case of hemp cells.
Okay.
So those two, playing with the nutrients, and the other one would be light. Those are the two easy ones, I would say. Now, in our research in our group, we actually introduced electricity as a form of elicitor, and we successfully demonstrated that. And I cooperated with that with a plant science professor. And both in cells as well as organs and whole plants, by subjecting them to low levels of electricity, whether it's alternating current or direct current, and of course we limited the amount of time exposure as well as the magnitude of the current, we were able to elicit secondary metabolites from plant cells that we tested. So I would say that would be applicable as well to hemp cells, but that would be down the road. We'll start with the simple elicitors like nitrogen or nutrient deprivation and manipulating the light intensity and light quality.
Sure, sure. I remember that elicitation method, Joel. When I first joined actually UFA back in 2007, we worked together with you also in the deep water culture system, trying to elicit some of the leafy greens and
oh, that's right,
and some of the secondary metabolites. That was an interesting research. All right. Other questions. Let's see. I think this is about elicitation. So the question is, where would the current be applied? To the plant itself or to the water or nutrient solutions?
to the liquid medium, to the nutrient solution.
All right, to the nutrient solution.
And it makes sense because, you know, the nutrient solution, there's cations in there, there's anions.
Mm-hmm.
And so once you have your electrodes immersed in the liquid nutrient solution and you apply the current will actually run through the liquid medium. And the cells will be exposed to the electrical current.
Okay. All right. All right, let's see if we have other questions. A follow-up question on this. Then what would this throw off the balance of the nutrient solution?
Okay, by balance, I'm assuming that would be the concentrations as well as the types of nutrients that will be available or present in the nutrient solution. So the quick answer is yes. I mean, certainly there are, okay, so it depends, depending on the plant species of the cells, as well as the particular metabolites that you are eliciting. Some metabolites are intracellular, so even after exposing them to these stressful conditions, they will overproduce the metabolites, but they will remain inside the cell. So those are called intracellular metabolites. So in the case of intracellular metabolites, they're not going to impact the composition of the nutrient medium, the liquid medium, because they remain inside the cells. But there are secondary metabolites that are extracellular, meaning to say that when they are overproduced, they also exit the cells and they go into the liquid medium. So in that regard, you're adding them into the liquid medium, and for sure, they're going to change the composition of your liquid nutrient solution.
All right. Okay, there was a link provided by one of our audience and colleague, actually, AJ Bood. It's a web link. I'm not sure what information is presented in that link, maybe related to the question or the discussion that we're having right now. But that's in the chat window for our audience. All right.
How many participants do we have?
Right now we have 26, Joel, but we started your presentation with about 42, I believe. Mm-hmm.
All right.
Yeah, it was a great participation again. We're slowly approaching to our time to end. Before we kind of conclude, I would like to thank everyone joining, participating for today's seminar. And I also would like to thank Rafi Gruner, our program coordinator for the seminar series, as well as Dave Bogner, our director of cyber systems and communications, and Ellen Worth, our program coordinator at the Controlled Environment Agriculture Center. And our speaker, Dr. Joel Cuello. Thank you, Joel, for this very nice presentation and discussion. I'm sure our audience and participants would follow up with you, and if they have any questions, I believe they can find you. We have your contact information also in our seminar announcement document. Please follow us for the upcoming seminars. This was our last seminar of the spring semester, but we will have seminars also during the summer period. So please follow us with the emails and in the website, CAC website. So with that, I would like to thank everyone. Have a great weekend, and please stay safe and healthy. And I'm gonna give the word to Rafi before we close, if he is still there.
I just wanted to add my words of thank you to, obviously, Joel, who once again provided us with a very thought-provoking seminar. Again, Dave Bogner makes it all possible. I don't know how he does it, but that's his expertise. He's our tech guru. and made this Zoom presentation not only easy, but flawless. Thank you, Murat. And I think Jean is also online. And so Jean was the one under whose leadership this series began. And so everybody stay healthy, and we will see you in the fall.
All right. Thank you, everyone.
Thank you, Rafi. Thank you, Murat, and thank you, Dave Wagner.
Yes, thank you.